Welcome to Know Your Mutation, Why is Biomarker Testing Important in GIST? During this seminar, all participants will be in a listen-only mode. There will be a questions and answer session at the end of the presentation using questions submitted through the Q&A feature. Please remember, the information provided in this web seminar is not intended as a substitute for your physician's guidance and care. My name is Christina Oakes, Data and Research Manager at the LifeRaft Group. I would like to thank Blueprint, Novartis, Genentech, and Daiichi Sankyo for their educational grants for today's webinar. I would like to introduce the presenters for today. Dr. Calvin Chow is the Senior Vice President of Medical Affairs at Tempus AI. Previously, Dr. Chow spent 13 years leading studies in education for the Oncotype DX breast, prostate, and colon cancer assays at Genomic Health, later acquired by Exact Sciences. Prior to that role, he worked various biopharmaceutical medical director positions at Onyx Pharmaceuticals, PDL Biopharma, TAP Pharmaceuticals, and SmithKline Beecham. In his early career, Dr. Chow practiced as a board-certified internist and hospitalist at Kaiser Permanente in California for 10 years. He earned his MD from the University of Texas Medical Branch, completed his medical residency at Presbyterian St. Luke's Medical Center in Denver, Colorado, received his MBA from St. Mary's College, California, and Bachelor's of Science in Chemical Engineering from the University of California, Berkeley. Michelle Harris, PharmD, MS, MBA, is a Senior Director of Medical Affairs at Tempus AI. Michelle has worked in the oncology space for almost 20 years in research, development, and medical affairs. She has been at Tempus AI for 2.5 years, leading the field medical liaison team who work with physicians on research and education. Prior to Tempus, Michelle worked in solid tumor drug development and clinical trials at Novartis and Gilead Sciences. We are going to start our webinar with a brief presentation from my colleague, Tony Powells, Research and Outreach Manager at the LifeRaft Group. Tony is a vital member of a team of patient monitoring coordinators and data analysts who provide day-to-day support of LRG patients. She participates in direct patient contact, including outreach, medical updates, email community support, and patient education, deploying her knowledge of medical information related to GIST, as well as educating and reassuring patients and caregivers. Tony also supports a development and involvement team planning educational events and fundraising events for our community. So, Tony, if you want to get started.
Hi, everyone. Thank you for joining the webinar today. I just wanted to give a brief introduction as to why biomarker testing is important in GIST treatment. So the first thing to say is I get a lot of patients asking if mutational testing, biomarker testing, tumor profiling, neogenomic testing, if they are the same thing. And these terms may vary slightly based on the specific context or focus of the testing, but they generally relate to the analysis of biological markers to understand what's driving your GIST and to inform the best treatment options for GIST patients. And there are many different reasons why mutational testing, or also known as biomarker testing, is important. One being, in some cases, biomarker testing can help inform or confirm the diagnosis of GIST by identifying specific genetic mutations. The most common mutations, which I'll be describing in the next slide, is the KIT gene, specifically KIT exon 11, and specifically the first-line treatment Gleevec is most effective for this particular mutation. In addition, it does help with treatment decision-making. The presence of specific mutations can help guide treatment options. Like I said before, the most common mutation, KIT exon 11, effectively responds to Gleevec, but in some cases, you might have a PDGFRA mutation, for instance, an exon 18 mutation, and the best drug that responds to this type of mutation is Ayvakit. And then moving forward, biomarkers can also provide information about the prognosis of the disease. For instance, specific mutations may correlate with the tumor aggressiveness or the likelihood of recurrence happening. And likewise, it might help to indicate whether or not there is resistance over time. For instance, you may be taking imatinib and there may be progression down the line, and getting further mutational testing can help understand whether or not there is a resistant mutation that is causing you to not respond to the imatinib. So it's also important to get testing further along in your medication journey as well. And then lastly, there are many active clinical trials recruiting, and many of these trials offer testing that can help identify whether or not their treatments are most effective for your treatment profile. So going more in depth, this image, it shows the mutational landscape of GIST. It emphasizes the mutation variability. As I said before, exon 11 is presented as the most common mutation, and the runner-up is KIT exon 9, and then there's PDGFRA and wild type that cover a small percentage. And within these mutations, there's something called codons. I like to think of the actual KIT exon 11 mutation as a section of instructions, and each codon is like a step in the instructions. For instance, within KIT exon 11, there's a codon 557 and 558, and these codons are the steps that are driving the mutation. And in some sense, these codons can help identify the aggressiveness or the responsiveness of the particular GIST mutation. So I just wanted to go more into our data at the LifeRaf group. This table is data from our 2023 data set. Just generally speaking, those who join the LifeRaf group are more likely to be connected with the resources and information needed to understand that mutational testing is important. So that's why we see that 70% of LRG members have gotten mutational testing compared to the global testing rates, which are below 30%. And this is over twice the global average. It may be because we're very good at advocating to get testing through us. And also those who join the LifeRaf group are more likely to be connected with a LifeRaf group GIST specialist who is more informed that mutational testing is important off the bat, and it helps to understand what mutation is most effective. And this is why we see that those LRG members with mutational testing have a higher median survival because they are on the correct medication for their mutation type. And then going more generally into the genomic distribution, this is the number of tissue samples we collect within our LifeRaf group tissue bank. We do have many collaborations. Earlier on, we collaborated with OHSU to get patients mutational testing, and more currently, we are working with Bayer Pharmaceuticals to get mutational testing through Tempus Labs. Aside from that, patients do just generally report their mutation status, and they get it through their local oncologist or GIST specialist. And we do collect the general GIST mutation information, and it's not a surprise that we do have mostly KIT mutations reported in our registry. And lately we have been also reporting additional biomarkers that may play a relation with GIST mutations. We've been collecting more data on that to understand the relationship. So now I wanted to go more in depth with how to join our free tissue testing project. This project started in 2021. We are in collaboration with Bayer Pharmaceutical, And this collaboration, it's an example of the vital role patient advocacy groups play in bridging the gap between researchers and patient populations. And our goal is to enhance access to mutational testing for all GIST patients. Within our initiative, we do have qualifications. One is that you must reside in the United States. Another is you must be a member of our patient registry. If you don't know what our patient registry is, it's an online database where we collect just research to enhance our just research for the populations. And we do require you to fill out a digital questionnaire. We like to collect this data so that if we were to get your mutation tested, we can analyze the patterns in your mutation and how it relates to your GIST tumor profile. And then also a requirement is that you have not gotten any type of prior mutational testing. So we specifically test patients who have not had any mutational testing before. Some patients, they do get the basic KIT or PDGFRA testing. And unfortunately, if you did get that testing already, we would not be able to do the testing. So it's solely for patients who do not know their mutation at all. Or if you have gotten the basic c-KIT or PDGFRA testing and it came back as no mutations detected, that places you under a wild type status. And we do offer further testing to understand what wild type mutation may be driving your GIST. And then this slide just outlines the process on how to get mutational testing. So the first things first is to reach out to us, whether it be through email or phone. We're always here for you. Next is signing up for our patient registry, like I said before, and I can walk you through that process. It's a matter of filling out a patient registry questionnaire along with an IRB consent, which gives us permission to store your information in our database. And then once we have your data for patient registry, I will be able to send out 2 consent forms. One gives us consent to request a tissue from your hospital where your tissue sample is at. Some patients ask if their tissue sample is still there. Typically in the United States, tissue samples are stored for 10 years, so it shouldn't be a problem. And then consent form B is obtaining your doctor consent. It can be your treating oncologist or treating surgeon, but we just require that you sign off on this so we can have them in the loop of what you're doing. And then once we have these forms completed, and just one thing to note is that with collaboration with Tempus, we would like everything de-identified. So please do not provide any information or such as your name on the consent forms. So once we have these forms completed, I or one of my teammates will be reaching out to your treating physician to get their consent and to request your tissue from your lab at the same time. And it does, time varies with this because sometimes it does take a while to get the signature from the physician, but once we do receive all materials, we will send out your tissue for testing. As you can see in the GIF, I am assembling the Tempus kit package. And once I assemble it, I'm able to send it out to the Tempus labs. And once they have completed testing, they will alert me and your treating physician, and I will alert you regarding your results. In some cases, your results may come back as a wild type mutation, which is, for instance, SDH or NF1. And if this is the case, we do offer further genetic testing to understand if you have any germline testing, germline mutation, which means that if it was passed down through family lines. But if you want to know more about that, I'd be happy to explain it more if you come across that bridge. And then I just wanted to give a sample of what a Tempest report looks like. I find them to be very informative because not only are your mutation is being reported, but it does show recommendations for potential clinical trials that you can join. And also it does display the variants of unknown significance, like I explained in the beginning. And then that's everything I wanted to cover. If you do have any other questions, you can ask at the end, or feel free to email our department. Thank you.
Thank you, Tony. That was a great presentation. So now we can move on into our next presentation by Tempest.
Sounds good. How did the slides, did they come up okay?
Yes.
Okay, perfect. Well, Antoinette, thank you so much for that great introduction to biomarkers and just because I think it's a great kind of way to segue into what Calvin and I will be talking about next, which is the comprehensive portfolio of Tempest and how we offer all of these different testing solutions and how they play into treatment of GIST, which I think is very important considering there's targets and there's targeted therapies that can really drive the right treatment to the right patient at the right time, which is what we're all looking for to improve outcomes. So nice to meet you again. And it's great to be with this group today. And it's great to be with all the patients that are listening or caregivers or providers. So thanks. It's an honor to be here. And we appreciate this forum to be able to talk to you. So within this presentation, we are going to talk about really Tempest, and we're going to give an overview. And I think Antoinette did a great job, but just another overview of the role of NGS testing in GIST, the impact it has on treatment choice, and then again, really focusing on Tempest and what our testing does and kind of how it plays into the treatment paradigm in GIST. So as we heard, next generation sequencing is very important. It helps to identify mutations or alterations. And why that's important is we want to understand what's going on in the tumor. There can also be things that are going on genetically, as Antoinette said, that's in your germline, which is all the cells you're born with. But really, we want to understand what exactly is going on in this tumor. Not all GIST tumors are the same. So we want to look at your unique tumor, what's going on with it, what mutations can we find in there so that your doctors can really understand the best treatment choice for you as you move forward into the treatment paradigm. So they're critical for guiding best treatment for each individual patient. That's what we call personalized medicine. We don't want to catch all, just give you what everyone else is giving you. We want to give you the treatment that matches up best for your tumor and what mutations we're seeing. And then we also want to understand how those mutations can correlate with your prognosis, because we know some patients do better on certain treatments, some patients don't. So what are those mutations telling us about you as a patient and how you might respond and what your prognosis is? So that backup plans can be made. So you can say, okay, we're going to try this, next we'll try that. All of that information is very helpful to know up front. We also know that outside of NGS, although NGS is very comprehensive, there may be other tests such as immune histochemistry that might be indicated. And that just helps to confirm what we're seeing on the NGS results. And it's just an additive test that may be required to just confirm and make sure we're really understanding what's going on. So you may hear that as well, IHC. So at Tempest, there's kind of a paradigm, and at the center of this is the patient. It's what can we do to really best help serve the patients and the physicians that are treating them. And so what we really want to know is what's going on in your tumor now. So as I mentioned, kind of right at this point in time, what is very unique and special to your tumor that we need to know so that we can understand what treatment would best align to that. We also want to know what other factors outside of your tumor do we need to take into consideration. So this is germline, what we are saying. What DNA were you born with that we're seeing in every cell in your body, not specific to your tumor, that we might need to take into account when we're treating you, or that might be important for your family members because that genetic material gets passed down, does that come into play for other types of cancers, or how you might respond to a treatment? So it also, there are certain germline or genetic codes that make you more vulnerable to toxicities of certain drugs. So we also want to know that up front, so that if that drug gets prescribed, we can really manage those toxicities or eliminate them. And knowing that up front is going to be extremely helpful, so that each patient has its best response and limit the toxicities as much as possible. We also want to know, and Antoinette referred to this, resistance. We want to know what's going on with your tumor over time, because tumors are kind of like viruses. They can mutate and change over time. And so when you select the best therapy, your tumor may respond beautifully, but over time it gets a little bit smart and starts to mutate, and other mutations may pop up, and we want to squash those mutations. So that's what's called resistance. And we really want to follow that over time and kind of understand and make sure that we're giving you the best therapy at that time to ensure that you're getting the maximum response. And then all of this information at Tempus, what makes Tempus a unique company is we collect all of this information, de-identified information, and we try to understand it in aggregate so that we can continue to understand insights and potentially inform new treatment options for patients with GIST. So again, I know Antoinette alluded to this, but how do we understand new targets? And she mentioned PTEN. How can we understand how these new targets come into play and these new mutations, so that we can develop new drugs and have new therapy options for patients as we better understand the tumor and how it evolves over time? So, of course, all of these things are taken into context when we're looking at the patient and really trying to understand, how do we treat you best now? And then hopefully, how do we develop new treatments for the future that are impactful to patient care? With that, I will turn the next part of the presentation over to Calvin, my colleague.
Great. Thank you, Michelle. Hi, everyone. So I'll be talking a little bit more about some of the clinical decisions for treatment and the specific biomarkers within GI stromal tumors, and a little bit about the therapies that are available. So the first thing I want to describe is some treatment concepts that relate to when the diagnosis is first made. If the tumor is actually either small or potentially shrinkable, to help spare the type of surgery that can be done. Neoadjuvant therapy, which means simply giving some kind of systemic treatment, some therapy before surgery is actually conducted, is often used to try to shrink the tumor so that we can really have a reduced need for as much resection of the tumor. And this really obviously is of high interest to patients, and it is oftentimes used with the therapies that we're going to be talking about. Depending on the specific biomarker in question, the specific drug would be used in that circumstance. There could also be a need for adjuvant therapy, which simply means giving the systemic therapy after surgery is performed to remove the tumor. This may happen if the tumor is felt to be of intermediate or higher risk, with some risk of recurrence, and giving adjuvant therapy really allows the treatment so that we can reduce those risks for the cancer of coming back. And then in the last bullet point, recurrent or metastatic disease is of course, in situations where a tumor, once resected, comes back, or patients simply have disease that is affecting more than just the primary site in question. Okay, next slide, please. So the first principle I'd like to kind of just review is that we're talking here mostly about biomarkers. And the biomarkers that you see listed in the different colors are KIT, PDGFR-A, and SDH, or those are probably the top three that are the most common and affect most of the GI stromal tumors in question. There are very small minorities of the others, KRAS, NF1, and others. I think that it's important to note that GI stromal tumors can occur in the stomach, the small intestine, or even in the colon and rectum. 2/3 of these are in the stomach. About 20 to 35% in the small intestine and a small percentage in colon or rectum. But even within the stomach, you can see that dependent on the location of the tumor, different patterns of biomarkers may be found. In fact, if we find tumors in the proximal part of the stomach, the fundus, you can see that almost all of these, more than 96% or so, actually have KIT mutations. And as you go further down in the stomach, the farther you go, the lower the rate of the KIT mutations and the higher the rates of other mutations, such as the PDGFRA and SDH. When you're in the antrum, which is the farthest distal part of the stomach, you can see that a little over half are PDGFRA and about a quarter is KIT. So I think this is important to know because sometimes even before, certainly testing is recommended in all patients, and I think that's always a key important piece of information to really help the treatment decision. But it does take a couple of weeks of time, and sometimes patients may find that they need to have their therapy started, especially if it's a neoadjuvant therapy, before you actually have those test results back. And so this kind of pattern of how frequently you find different mutations can be a helpful guide, because if we're talking about a proximal tumor, sometimes it's very likely to be a KIT mutation, and the imatinib could be started in a neoadjuvant context, pending confirmation of what the mutation actually is. This is based on what is stated in the guidelines. But I think as the farther you go in this location in the stomach, we have to acknowledge that some of these other mutations may be at play, and we know that imatinib is actually not as effective in some of the other alterations. So we'll talk about that shortly. Next slide. Oops. Could you go back one? No, there we go. Sorry for the small text here, but I'll talk through some of these points very carefully. The first is that, as you saw from the earlier slide, about 80% of GI stromal tumors will have a mutation in the KIT receptor tyrosine kinase. And there's an interesting history here. The discovery of KIT dates back to about 1998. And imatinib is actually a drug, Gleevec. It was used originally found for CML, chronic myelocytic leukemia. And the approval for imatinib for KIT-associated GI stromal tumors actually occurred in 2001, FDA approval. So it's been around for some time, and a lot has been learned about this alteration in the subsequent years. And we'll talk a little bit about resistance and the emergence of some of these other treatments. The second most common mutation is the PDGFRA mutation. And here, this represents about 5 to 10% of all GI stromal tumors. This is actually, there's a mutation in exon 18 of with the D842V variant. And these are the most common PDGFRA alterations. And this is discovered and found that there was a primary resistance to imatinib, and it was the most common PDGFRA alteration. And so this led to the development of avapritinib, which was FDA approved in the year 2020. And so I think between these two genes, these are all targetable initial therapies that can be offered, and these are both TKIs. And I think that's important to know that these really represent the two preferred initial regimens for GI stromal tumors in general. Mutations also confer some prognostic information, as was said earlier, and this really can give insights into what is the likelihood of response, as well as what is the likelihood of recurrence or potential resistance to standard therapies. The next statement is that there are obviously some tumors that are resistant or that don't have either of these mutations in KIT or PDGFRA, about 10 to 15%. And the most common one of these is the SDH mutation or alteration. SDH stands for succinate dehydrogenase, and we kind of refer to the deficiency of SDH as the primary alteration. It could be due to a mutation in the gene itself, or it could be due to something that's not a mutation. Sometimes there's a condition called hypermethylation, or it's what they call an epigenetic process that results in the deactivation or the decreased expression of that gene. And this is detected, therefore, by IHC testing, immunohistochemistry testing, for this SDH-B non-expression. And now these tumors tend to be less responsive to the drugs that we've just talked about, imatinib and tyrosine kinase inhibitors, although they are potentially responsive to sunitinib and regorafenib. So it's important to do testing, therefore, with not only the next generation sequencing test, but also with immunohistochemistry to detect SDH. These alterations can arise, the SDH mutations and alterations can often arise in younger individuals. Again, these are typically resistant to imatinib, but they may respond to sunitinib or regorafenib, as said earlier. There could also be an association of patients who have germline SDH mutations with a risk of paraganglioma. And so this might infer that there might be some importance to germline testing and workup for this. The guidelines also have mention of other potential drivers that are much lower frequencies: NTRK fusions, BRAF mutations, NF1, neurofibromin 1, and FGFR fusions. And tissue biopsy is typically preferred for that initial diagnostic biomarker workup, although circulating tumor DNA may be appropriate in some cases as well, and may be also conveniently used in subsequent time points when you're looking for emergence of resistance mechanisms. And then finally, the NF1 gene, those alterations typically arise in the small bowel, the small intestine. These tumors tend to be, it's related to neurofibromatosis, the condition, and this can be a genetic condition. And so if found, genetic counseling may be warranted, or further genetic workups, if not previously undertaken. And these tend to be more indolent, but may be multifocal and present in a variety of locations throughout the small intestine. Okay, next slide. So here is a list of the relevant biomarkers and their associated therapies in first line and in second line settings. Once again, KIT, the most common mutation in GIST, and PDGFRA are the number 1 and number 2 in terms of frequency. I think it's important to note that imatinib is typically indicated for the preferred therapy in the first line setting. However, if a patient has the PDGFRA mutation in exon 18, if it's that one particular mutation called the D842V mutation, this would then be resistant, not typically responsive to imatinib, and one should receive avapritinib that was approved just in 2020. And you can see in the second line setting, once patients actually develop resistance or have a new progression on these therapies, you can see the therapies that are indicated in the second line or later. A lot, again, has been learned about these tumors. It's very common that patients may, over a course of time, it's important to note that imatinib therapy can go on for quite some time. The median overall survival for that therapy, when it first was approved, increased the median overall survival from 12 months to about 4 to 5 years. So it's really quite a substantial increase in outcomes from just that first therapy. But it is known that as time goes on, there could be some degrees of resistance that emerge. And initially, there is a potential quiescent state or kind of a non-proliferative state that these tumors can go into that simply represent just a potential resistance mechanism that's there. But eventually what happens is that these persistent clones can eventually acquire another mutation. And so these mutations then emerge, and that would then signal the need for potential switch to other therapies. So here we have at second-line therapy, it's mentioned that sunitinib here is the preferred regimen for the second line. If patients then progress on that third-line therapy, the preferred regimen is regorafenib. And then upon progression there, there is a newer agent called ripretinib. In the fourth-line setting it is approved for that fourth-line setting in that circumstance. And so this is something where you can see that really as patients continue to evolve with these therapies, this may be the sequence of potential therapies that are received by the patient. You can see that in the PDGFRA exon 18 circumstance, where avapritinib was the first-line therapy, you can see second line could be the dasatinib upon progression. Below the lower part of the table, there are some of the rarer gene findings, NTRK fusions, the SDH deficiency found by immunohistochemistry, and BRAF V600E. And you can see the associated therapies here. There are some NTRK inhibitors, entrectinib, larotrectinib, or repotrectinib. For SDH deficiency, sunitinib is the first-line therapy that's preferred, followed by in second-line regorafenib, pazopanib, and imatinib, as you see here, potentially with binimetinib in a combination. And then finally, BRAF V600 is typically managed with dabrafenib and trametinib. Okay, next slide. So a lot of different potential molecular findings to look for and generation sequencing. This is a busy slide, but I think I want to point your attention to the third column test there called XT. This is the solid tumor DNA sequencing that's offered by Tempus, where 648 genes are looked for for DNA alterations. This is oftentimes coupled with the next test over on the fourth column, XR. XR simply represents the whole transcriptome RNA sequencing, where the RNA can be important in finding gene fusions, for example, like the NTRK that we talked about, where this can be detected a little bit better with RNA than with DNA. And so oftentimes doing these together from that same tissue specimen is important. And so I would say that most of these represent the typical initial testings with tumor tissue next generation sequencing. Liquid biopsies represented over on the far right under XF and XF plus represent the plasma CT DNA sequencing, circulating tumor DNA. These are two tests. One has 105 genes and the other 523 genes. And this can be helpful if tumor tissue is either not available, or the sequencing fails in tissue, or tissue is very scant. It can also be done at the same time as the solid tumor tissue sequencing, because we do find that sometimes liquid biopsy will capture something that was not found on tumor tissue, and vice versa. Sometimes tumor tissue finds things that liquid biopsy does not find. And so there is a convenient auto conversion opportunity where sometimes the physician may order both the tube of blood with the sequencing of the tissue. And if that tissue didn't succeed, we can actually just auto reflex or convert that tube of blood to do the liquid biopsy XF or XF plus. Lastly, there is a XG and XG plus hereditary cancer panel test with 36 and 77 genes to work up potential hereditary cancer syndromes. This is coupled with the availability of genetic counseling and familial variant testing, meaning that if a patient is found to have an alteration that could be genetically important, that family members can also be tested. And I won't mention too much about the test down at the bottom, because I think this goes beyond the scope of the GI stromal tumor diagnosis here. So with that, we can conclude. Thank you for this opportunity, and thank you all for listening. Appreciate it.
Thank you.